Hello, we're starting the second part of Blood Flow 1, and this is on filtration and absorption. So we know that fluids can leave a vessel, so we're thinking of a capillary, can leave a capillary by bulk flow. It results, I'm telling you, from hydraulic or osmotic pressures. So what does all this mean? Imagine that capillary and imagine that it has quite a bit of pressure inside because blood is flowing. And so that pressure is going to make this leaky hose leak a little bit, right? Also, it has osmotic pressure because inside this hose is blood, and on the outside of the hose is a very thin fluid, watery fluid that's outside that vessel. So think of that, but we're going to come back to that concept. When something flows out of the capillary, we're going to call it filtration. And that's the soaker hose analogy. It's going to leak out of it from the blood over towards the tissues. That's going to be filtration. When it's coming back in, because that's going to happen, right? We're going to collect up carbon dioxide, for example, or wastes. Then we're going to call it absorption. So absorption means it's going back into the capillary. If we want to define it, just to be certain, we're going to define it as bulk flow refers to the mass movement of fluid as a result of hydrostatic or osmotic pressure. We're going to see a lot more about this, so if it's still not clear, don't worry. Think of it as fluid moving, carrying its solutes with it, because if this soaker hose is going to leak, it's going to take whatever's inside with it. So here's my picture of a soaker hose. If you don't do gardening, you can have a hose like this, and you turn the faucet on, and just the pressure from the water is going to make it leak out onto your plants, and it just will wet the whole big area. If you put on more pressure, more of it is going to leak. If you put less on, less it's going to leak. But along with it is going to come whatever is in that water. So if there's chlorine in it, it's coming out too, right? If there's any sodium in it, calcium deposits, it's going to come out through that hose. So this is a really nice analogy for what it means when your capillary is filtering. So think in the hose, my question one here, what makes it drip? What makes it drip is the pressure that it's coming with, right? If you really turn on that faucet high and increase that pressure, it's going to drip out more. And my second question, what drips out along with the water? Remember, it's whatever solutes are there are going to come with it. So this is bulk flow. Drippy capillaries are responsible. Defined again, it's a process used by small lipid insoluble proteins to cross the capillary wall. Capillary structure plays a large role in the rate of bulk flow. What's that mean? Capillary structure, so some of them are leakier, some of them are less leaky. So we've talked about the different types of capillaries. So with continuous capillaries, it's going to limit that flow. And with fenestrated capillaries, it's going to increase that. It depends on what part of the body they're in. They're going to leak more or less, and you need that. Remember that this excess fluid, if you're thinking, oh my gosh, it's leaking out, and then where's it going? Remember that the lymphatic system is going to be able to pick some of that back up again and put it back into circulation. So you're not going to have edema when this is happening, right? This is a regular mechanism that happens in your capillaries. Which capillaries are the ones that are going to absorb and which are the ones that are going to filter, right? Because we said sometimes it's going out and sometimes it's going in. Well, to think about that, you have to go back to the drawing. Let me get my pen. That we had before, where you have the capillary is going to be connected to an arteriole and a venule, and this is the capillary bed in here. And so most capillaries are going to do both. They're going to be able to absorb, and some of them are going to filter. So the net filtration, this is very interesting, is going to occur at the arterial end. So more filtration is going to be happening. It's going to go in both directions. Some is going to come in, some is going to go out. But really, the overall net filtration is going to be at this arterial end. You notice at the end closer to the artery. And the net absorption is going to be on the venous end. So can you think that why that is? First of all, think of it in terms of pressure. The pressure is higher here as it's coming from this arterial, and it's going to go dropping, dropping, dropping by the time it gets to the venule. So you have less pressure. The pressure is going to be lower as it travels here. It's going to be harder for that soca hose to actually drip out, essentially. Okay, but we'll see more about this. Where does it leak in or out of? So we got to remember here that it's through the endothelial cells. It can go right through the endothelial cell if it's fat soluble or very small. The examples of that are oxygen and carbon dioxide, and they can do that right through the cell, go in one end of the cell and out the other. So it's just like a sponge there. Now, endocytosis is used, remember, for larger molecules that can't just diffuse through. So that fluid volume is maintained by balancing these opposing forces at the capillaries. What were the opposing forces? The opposing forces are pressure, which is blood pressure, and osmotic pressure. And we haven't talked enough about that yet. The blood pressure is tending to push it out of the capillary, and the osmotic pressure is wanting to draw it back into the capillary. And why is that? Because you have a fluid, which is blood, which is full of solutes and of proteins, very different from the extracellular fluid that's just a watery substance. So if you have osmotic pressure, it's going to be in which direction? Remember, when you have solutes, water is attracted towards the solute. So if it's coming in towards the solute, it's coming in towards the blood or to enter that capillary. So this is driving it out. I think I might try and make a drawing of that for the osmotic pressure. Let's do that. So if you had a capillary like this, and in here is blood, and in here I'm going to put lots of solutes in here because there's proteins. We're going to make big proteins like this, and there's lots of salts, and on the outside here is a watery fluid, which is the extracellular, hard to write with this, extracellular fluid. So when we think about fluid, if you just, even if it weren't flowing and you were sitting with something that was very concentrated in solute here and not so concentrated outside of this membrane, and many of these solutes are not moving, what's going to happen is fluid or water is going to want to move in towards the higher solute. So osmotic pressure is pulling things into, wanting to pull things into the capillary. But if you have blood pressure, let me get in another color. If you had blood pressure that's huge here, we can make a pressure, which is a very big pressure coming out like this. Then this pressure is going to overcome the osmotic pressure. So at certain parts of that capillary, the blood pressure is, we're going to call it huge, and overcoming the osmotic. But as that blood pressure diminishes down as it flows, and we're going to make this, my arrow getting smaller here on this end, then at some point the osmotic pressure may overcome that the blood pressure. We call it hydraulic pressure that's driving the fluid out of the capillary, because hydraulic means fluid in motion, but it's really the same as blood pressure. That's what we mean. Colloid osmotic pressure. It's helpful to think of solutes suck. And there's my drawing again. It's going to drive that fluid back into the capillary. And it's mainly because of those plasma proteins and solutes that are within the blood. It's the same as oncotic pressure. If you see that in the book, referred to as oncotic pressure, it's the same, colloid osmotic pressure. So this is bulk flow happening. So take a look at this diagram that illustrates this very well. You have a capillary here, and you have a red line that's showing how the osmotic pressure changes from the arterial end to the venous end. So how does it change? It doesn't, because you have these large proteins, and that pressure, the osmotic pressure, is about 25 millimeters of mercury on this end. And as you keep traveling to the other end, it's still 25 millimeters of mercury. So that is not changing. That's staying constant. What is changing is the blue line here, which is the blood pressure. It's this arterial end is very high, and as it travels along, therefore, there's going to be net filtration, because that pressure, that blood pressure, is pushing blood out. It's overcoming the osmotic pressure. But at a certain point, look, here they're equal. And then beyond that, the osmotic pressure is now higher. So at this end, at the venule end, net absorption is what's going on in this capillary. So all capillaries are like this. If you have this long capillary bed, always at the arterial end, there's going to be more filtration. And at the venule end, there's going to be more absorption. So that works well, because you're distributing oxygen, for example, on this end, and you're picking up CO2. You're picking up CO2 at the other end. This is a really good diagram, so stare at it for a long time. I think we're going to stop this there. We're going to stop this mini lecture there and start the next one with the next slide. Thank you for listening.